Angelman Syndrome Genotypes, sponsored by the Foundation for Angelman Syndrome Therapeutics. Everyone inherits their chromosomes from their parents. The chromosomes carry genes, which are coding instructions for making proteins. An important example of this is the UBE3A protein, which is made from the UBE3A gene located on chromosome 15. People living with Angelman syndrome, or AS for short, do not have enough functional UBE3A protein in their brains. Typically, each person has 2 copies of the UBE3A gene, one from each parent. The one from the father is called paternal, and the one from the mother is called maternal. In all people, the paternal UBE3A gene is silenced or turned off in brain cells, a process called imprinting, so the maternal UBE3A gene is responsible for making all the UBE3A protein in the brain. Now, changes to the maternal UBE3A gene can result in less or no UBE3A protein in the brain, or the UBE3A protein that is produced may not work properly. These genetic changes are grouped by type and are called genotypes. There are 5 main genotypes that cause AS. Knowing the genotype of each person living with AS is important. Although each individual living with AS is unique, people with the same genotype may have certain behaviors or characteristics in common. Another reason for identifying the genotype is that some genotypes have a higher chance of running in families. Doctors and genetic counselors can use this information to determine the chance of other family members inheriting AS. Finally, genotype is often a criteria for participating in research, so knowing the genotype is a key element in determining eligibility for clinical studies investigating potential AS therapies. Imprinting center defect, or ICD for short, occurs in about 3% of people living with AS. Normally, the imprinting center's job is to control and reset imprinted genes, like the UBE3A gene. However, when there is a defect in the imprinting center, the UBE3A gene may be incorrectly turned off in the brain, even though the UBE3A gene was inherited from the mother and is supposed to remain on and active. ICD can be caused by either a deletion of the imprinting center, called ICD deletion AS, or an error that affects the function of the imprinting center, called ICD non-deletion AS. In ICD non-deletion AS, the imprinting center does not have any detectable sequence differences, but is still not functioning correctly, resulting in the maternal UBE3A gene being turned off when it should be on. ICD deletion AS can be inherited from the mother if the mother also has that same deletion, and any future pregnancies will have a 50% chance of having ICD deletion AS. It may also be a de novo, or new random event that occurred during the formation of only one egg in the mother. On the other hand, ICD non-deletion AS is believed to happen randomly, often after conception, and is not typically inherited. For more information about Angelman syndrome and its genotypes, visit the Foundation for Angelman Syndrome Therapeutics, also called FAST, at cureangelman.org. For specific questions, reach out to FAST's genetic counselor.